Good morning students. Today is lecture 2 under module 11, and as you know that in this module we are discussing the various organic commodity chemicals that can be produced from biomass, and in today's lecture we will cover essentially 3 of them: succinic acid, propionic acid and acetic acid as well as butyric acid. So, let us begin. First we will discuss about succinic acid. Succinic acid, a 4 carbon dicarboxylic acid, is one of the end products of anaerobic fermentation and is also an intermediate of the TCA cycle, that tricarboxylic acid cycle. Now, various important chemicals including 1,4-butanediol, tetrahydrofuran, gamma butyrolactone, N-methyl 2-pyrrolidone and adipic acid can be synthesized from succinic acid. When the US Department of Energy identified succinic acid as one of the 12 promising fundamental chemicals to be produced through biotechnology in 2004, it was suggested that bio-based succinic acid will become competitive with the petrochemical process only if its productivity approaches 2.4 grams per liter per hour. So, this is the target they have faced, because some sort of techno-economical evaluation they have carried out and found out that if it is more than 2.5 grams per liter per hour, the productivity of the succinic acid from the biological materials, then it will become competitive. A number of succinic acid producing microorganisms have been isolated or developed together with the fermentation and recovery systems. Escherichia coli and natural succinic acid producers such as Anaerobispirillum succinoproducens, Actinobacillus succinogens, Mannheimia succinoproducens have been metabolically engineered to achieve higher yield and productivity. Many available and cost competitive carbon and nitrogen sources have been investigated for each succinic acid producers for the efficiencies of growth and succinic acid production. In addition, various mixed and semi purified carbon and nitrogen sources including the cane molasses, corn strip liquor, soybean flour, wood hydrolysate, yeast extract, polypeptone as well as crude glycerol, mostly from the biodiesel production industry, have been examined for succinic acid production. In parallel with the development of the fermentation process of succinic acid production, several different downstream processes for the separation and purification of succinic acid from culture medium have also been investigated and developed. For example, ultrafiltration was used widely for the removal of cells and cell debris as an initial separation stage. If you recall, even our yesterday's discussion, I was giving more emphasis that when we produce certain commodity chemicals using the biotechnological routes and even if thermochemical routes also, once they are produced, they will be mostly in the aqueous phase, then you have to carry out the downstream separation part. That means that is basically purification, right. So, usually 7%, 10%, 15 to 20% will be the initial concentration of the commodity chemicals in the fermentation broth or in the aqueous medium when you talk about thermochemical conversion, right. So, you need to purify it to up to 70%, 80%, so that you can use it as a bulk commodity chemical. So, here that is the catch. So, the downstream processing part is supposed to be very costly, still it is very costly. With the invention of membrane and all these things, process cost has coming down slowly and more work is still yet to be done, and people are doing day and night so much of research on this particular aspect, that how to develop low cost as well as sustainably economical downstream processing part for purification of commodity chemicals, okay, when once they are produced. A membrane has been tried a lot. Apart from membrane, there are other processes, and we have also discussed that how hybrid processes has also been taken into account and been successful for certain chemical purification. So, additional steps in the downstream processes including precipitation, reactive extraction and electrodialysis have been developed to selectively separate succinic acid from the fermentation mixture. Ion exchange and crystallization procedures were adapted to further purify and crystallize succinic acid. We will talk about some of the carbon sources from where succinic acid can be produced. Various kinds of carbohydrates are metabolized to succinic acid by diverse succinic acid producers. A succinoproducens is able to catabolize several carbohydrates that include glucose, sucrose, fructose, lactose, soluble starches as well as corn syrups, while M succinoproducens can utilize sucrose, fructose, lactose, xylose, maltose, mannitol and arabitol as efficiently as glucose. Now, moreover, many kinds of carbohydrates including glucose, sucrose, fructose, lactose, arabinose, galactose, maltose, mannitol, mannose, xylose, soluble starches and corn syrups can be used for succinic acid production by a succinogens. Further, more reduced carbon sources such as glycerol can be utilized by A succinoproducens and M succinoproducens. in the fed batch fermentation of egg succinic producents with glycerol as the carbon source, high succinic acid yield of 1.6 gram was achieved with a low accumulation byproducts. Now, in the same manner, by using the sorbitol as a common source, succinic acid yield was increased compared to glucose in using E. coli. And the reason is that glycerol and sorbitol are more reduced than glucose and provides more reducing power. In another study, employing carbohydrates using a transport system other than the phototransferase system was demonstrated to increase the succinic acid yield, because more carbon flux could be directed towards the oxaloacetate formation at the PEP node. In addition, co-fermentation of fructose, xylose or galactose with glucose was studied for E. coli as well as A-succinic producents by various researchers. Okay. Some of the references are given. You can if you are interested to learn more, you can please browse through these particular references. These are classical works. Instead of employing purified carbohydrates as carbon sources, some cost effective feedstocks such as wood hydrolysates, cane molasses, cheese whey and straw hydrolysates have been used for bio-based succinic acid production. In 2000 and 2003, Lee et al. Investigated an anaerobic fermentation process for succinic acid production from whey and wood hydrolysates by A-succinic producents. In the batch fermentation with 20 grams per liter of non-treated whey and 7 grams per liter of glucose, the yield and productivity of succinic acid of 0.95 gram per gram and 0.46 gram per liter per hour respectively were obtained. When wood hydrolysate, that is almost equivalent to 27 grams per liter of glucose, was used as carbon source, the succinic acid yield of 0.88 grams per gram was obtained. So, what you can see from this particular slide is that the succinic acid which is being reported is extremely low when you talk about the 2.5 grams per liter per hour productivity. Okay. However, these are classical works which has led a new path, or you can say open up new avenues to work on bio based succinic acid production, whereas we are now using genetically engineered strains to increase the yield, okay, as well as productivity. In the case of M-succinic producents, whey and wood hydrolyzed based medium were utilized as economical carbon sources to produce succinic acid. So, very important two papers which are given below, you can browse through them if you want to learn more. They are excellent work in this particular field. Then using corn strip liquor containing whey based medium, the succinic acid yield of 0.71 gram per gram and productivity of 1.18 grams per liter per hour was obtained in batch fermentation. Now, this is batch fermentation. Of course, when you talk about batch fermentation and compared with continuous one, it is always less, but it has its own advantages. Okay. Now, continuous fermentation resulted in succinic acid yield of 0.69 grams per gram and productivity of 3.9 gram per liter per hour. You just notice that the productivity is almost double, okay, 1.18 to 3.9, right, when we go for batch to continuous fermentation mode. Now, by using wood hydrolysates based medium, the succinic acid yield of 0.56 gram per gram and a productivity of 1.17 gram per liter per hour were obtained in batch fermentation, and 0.555 gram per gram and 3.19 gram per liter obtained in continuous fermentation. Again, you see that from 1.17 to almost double, 3.19 gram per liter per hour, the productivity is enhanced when we went for continuous fermentation using the wood hydrolysates. So, various studies utilizing the inexpensive carbon sources such as carbon fiber hydrolysates, cane molasses, cheese whey, wheat milling byproducts and straw hydrolysates by A-succinogens have been carried out to examine the possibility for cost effective succinic acid production. You will see that there are many, I could not report here also, just those were very important results we have listed here, right. So much of work you will see from succinic acid production using two very different approaches. First, different types of low cost biomass, okay, either it is low cost or it is almost no cost, right. So, just like waste, only you have to segregate and then little pre-processing and then you can use it. Second, using different types of strains, okay, mixed strains has also been reported, then genetically engineering strains have been reported. So, these are the two ways on which most of the succinic acid production research has been carried out, right. Now, let us talk about propionic acid. Propionic acid is a colorless naturally occurring carboxylic acid with a slightly unpleasant odor. It is soluble in water, organic solvents as well as alcohols. As a specialty chemical, propionic acid is mainly used as a preservative in food and grains. About almost 52% of what is being produced worldwide, bakery and dairy products 18% and in herbicide 16%, cellulose acetate propionate, okay, one of the another classical commodity chemical 10%. Now, sodium propionate can also be used in treating dermatoses, wound infection and conjunctivitis. In addition, esters of propionate are used in artificial fruit flavors as for example, citronellil and geranyl propionate, and of course, in plasticizers as for example, glycerol tripropionate and phenyl propionate. Propionate esters are also good substitutes for volatile solvents such as xylenes and certain ketones classified as hazardous air pollutants. In the past 10 years, the US market for propionic acid continued to grow at an annual rate of 2.3% with a sale price reaching $0.92 to $0.97 per pound in 2009. Current industrial production of propionic acid is mainly through the petrochemical process. Mostly it is from ethylene, carbon monoxide and steam, which is the REPE process, or ethanol and carbon monoxide, which is the well known Larsen process, and of course, both are carried out in the presence of catalysts. Now, due to the non renewable feedstocks derived from the increasingly expensive crude oils and environmental concerns of the petrochemical processes, there is a great interest in producing bio based propionic acid via fermentation using the Propionibacteria, okay, which are widely used in industry for the manufacturing of the Swiss cheese and vitamin B12. Okay. Propionic acid fermentation using renewable biomass such as agricultural residues and industrial wastes as feedstock is sustainable and environmental friendly. The bio-based propionic acid is also more appealing to the consumers for uses in foods and cosmetics. However, conventional propionic acid fermentation is uneconomical due to the low productivity yield, is very low 0.5 grams per gram, and the productivity is also less than 1 gram per liter per hour, and product concentration which is less than 40 grams per liter caused by the end product inhibition. So, let us talk about some of the propionic acid bacteria which are being used by various researchers even in the commercial scale to produce propionic acid. Many bacteria are capable of producing propionic acid. Okay. I am just showing you the table 1. You can see here some propionic acid producing bacteria and then general characteristics are given. We will come back to it later. Okay. Among them, Propionibacterium can produce propionic acid as the major product in the dicarboxylic acid pathway. Okay. Figure 1, here I will show you again what is this dicarboxylic pathway, the first one, and then acrylic acid path of the Clostridium species. Anyway, we will come back to it, okay, and is the most widely used genus for the propionic acid fermentation. Another species of interest is the Clostridium propionicum, okay, which can ferment lactate, alanine, serine and threonine, okay, to produce propionic acid and acetic acids via the acrylic acid pathway. Okay. The second pathway we will go to that. Okay. Some species in the genera Bacteroides, Fusobacterium, Megasphaera, Celomonas and Veliona can also produce propionic acid, but not all of them can use sugar or carbohydrate as carbon source. So far, propionic acid fermentation research has focused on Propionibacterium because of their better fermentation performance and broader substrate spectrum compared to other genera. Yes, this is the table 1. Please have a close look at this table. You can see that what are the different types of the species that are being used to produce propionic bacteria, whether they are gram positive, gram negative, what are the substrates they could degrade, what are the products, what are the optimum pH and temperature. So, we will just go 1 or 2, I will just tell you, rest you can see later on when you go through the lecture. The first is the Propionibacterium acidopianaceae, okay, and the class of Propionibacterium species. So, they are gram positive strains, they can degrade various types of substrates, whether it is glucose, sucrose, lactose, lactate, even glycerol also. Now, the product is propionate, acetate, succinate and carbon dioxide, of course, at different proportions. Optimum pH is 6.5 to 7, optimum temperature is 30 to 32, right. So, if you look at some other one, let us say the last one, the Fusobacterium necrophorum, it is a gram negative bacteria and it will only degrade lactate, okay. It is a lactate based or bacteria which will produce propionic acid. The end products are acetate, propionate and butyrate, again at different proportions. pH has to be maintained at less than 7.5, that is the optimum, and temperature is little slightly higher, it is 39, okay. Now, there are others also, you can go through it later on. This I showed you few minutes before. So, this is the classical propionic acid fermentation pathway, basically two different pathways. First is the dicarboxylic acid pathway of the Propionibacteria, second is the acrylic acid pathway of the Clostridium species, okay, Clostridium propionicum. So, let us try to understand what is happening in the dicarboxylic acid pathway, okay, of the Propionibacteria. So, you can see that here the substrate is glucose, okay, it can be something else also, right. So, glucose is getting converted to 3-phosphanopyruvate, okay, it is a pyruvate cycle again, then it is finally degraded to pyruvate. Now, this pyruvate can result into 3 different products, okay, again using different pathways. Now, when you talk about this particular pathway where the acetyl coenzyme A is getting produced, you remember this we have discussed in during fermentation also, okay, a little different, right. So, this pathway we have discussed. Now, that gives a intermediate product which is called acetyl phosphate, and that finally being degraded and to a end product which is acetate, right. Now, similarly pyruvate can go through 2 different other pathways. So, one is that oxaloacetate pathway, one is the methylonyl coenzyme A pathway. So, if you go through methylonyl coenzyme pathway, it will give is succinyl coenzyme A and finally 2-succinate, right. And here also if you talk about propynyl say methynyl coenzyme A, it will go to the propynyl coenzyme A and 2-propionate, right. Now, 2-methyl coenzyme A is also coming partly from the 2-succinate. Okay. Now, this 2-succinate is also getting converted in a backward cycle, right, to succinyl coenzyme A and then methylonyl coenzyme A. And if you look at the oxaloacetate pathway, say it gets converted to malate, fumarate. Okay. Now, this fumarate is getting converted to succinate. So, this is your dicarboxylic acid pathway of the Propionibacterium bacterium. Now, let us talk about the acrylic acid pathway of the Clostridium species. Now, here you can see that if we start with alanine, okay. So, alanine is getting converted to pyruvate. Now, pyruvate to acetyl coenzyme A, acetyl coenzyme A to acetyl phosphate to acetate. Now, pyruvate to lactate, lactate is getting converted to lactyl coenzyme A, then acrylyl coenzyme A, then propionyl coenzyme A and finally propionate. Okay. So, these are the 2 major pathways to produce the propionic acid. Okay. So, propionic acid production from the renewable feedstock. So, we will try to understand what are the biomasses that are being used. As the raw material cost usually accounts for a major portion of the final product cost in fermentation production of the oxychemicals including propionic acid, it is important to use inexpensive or low cost feedstock for propionic acid fermentation to make it more favorable to compete with the chemical synthesis route. There are large amount of wastes or byproducts generated from food, agricultural and other industries including cheese whey from the dairy industry, canned molasses and sugarcane bagasse from the sugar industry, corn fiber and CSL, corn strip liquor basically, okay, from the corn milling industry and glycerol from the biodiesel industry, which may be used as low cost feedstock for fermentation process. Now, these wastes containing fermentable sugars or carbon sources have low value, but high biochemical oxygen demand and chemical oxygen demand, the BOD and COD, posing an environmental threat if they are not treated properly. So, it is something you can say that waste to value added products. Okay. Thus, the application of these low cost and renewable wastes as an alternative feedstock to produce value added chemicals such as propionic acid has attracted large interest from both industry as well as academia. Some fermentation studies using low cost biomass feedstock are summarized in the table 2. So, you can see that the different types of substrates here, processes, the titer, the productivity, the yield and the respective references, right. Now, this table has been taken from this particular reference, you can browse through later on. So, let us just see 1 or 2 whey permeate, okay, a recycle batch with fluidized bed process, right. So, the titer is 65 grams per liter, productivity is 0.2 to 0.68, it is of course less compared to the theoretical one, yield is 0.5 grams per gram. Similarly, if you go to corn mill, this is a batch process with again a fluidized bed operation, okay, the titer is 40 grams per liter, however, the productivity is higher in this case 2.12 grams per liter per hour, okay, and the yield is 0.58 gram per gram. Similarly, if you look at some other things, you can see Jerusalem artichoke, lot of work has been done on this, sugarcane bagasse, glycerol, then glycerol and potato juice and respective productivities, okay, as well as the yield. The US dairy industry annually generated more than 80 million tons of whey as a byproduct from the cheese manufacturing process. The main components of cheese whey are whey protein and whey lactose, which makes up to about 75% of the whey total solids and are currently a large surplus of the limited uses. Propionic acid production from whey lactose and whey permeate by propionibacteria has been extensively investigated. In general, high productivity 0.22 to 0.68 grams per liter per hour and a yield of 0.5 grams per gram lactose with a high propionic acid concentration of 65 gram per liter can be obtained from the concentrated whey permeate. Calcium propionate produced from whey can be used as a natural preservative in bakery and dairy products. Corn fiber and corn strip liquor are two low value byproducts from the corn wet milling industry. They are currently used in mainly for the animal feed purposes, but with a large surplus because of the rapid expansion of the corn ethylene industry in recent years, they are being unused. Corn strip liquor is rich in amino acids and trace elements just like some of the metal ions and vitamins, and is a good nitrogen and nutrient source for industrial fermentation. A high final titer of propionic acid almost 46 grams per liter has been reported by using corn strip liquor medium in a continuous propionibacteria fermentation. In another study, Huang et al. Reported a high yield and high productivity almost 2.12 grams per liter per hour of propionic acid in a recycle batch fermentation process using the hydrolyzed corn meal as substrate. Molasses is a byproduct of the sugar refining industry and its major component is sucrose, glucose and fructose. Propionibacterium is capable of converting all these three sugars into propionic acid. A propionic acid fermentation process with non-treated or hydrolyzed molasses as carbon source reached a high propionic acid concentration of 92 grams per liter with a yield of 0.46 gram per gram and a productivity of 0.36 gram per liter per hour as reported by the Feng et al. In 2011. Okay. Respective references are given below. If you wish, you can browse through them later on. Sugarcane bagasse is another cheap and abundant byproduct of sugar refining, which can be hydrolyzed into glucose and xylose and then used as carbon source for propionic acid production, achieving a titer of 51 gram per liter, yield of 0.43 gram per gram and a productivity of 0.71 gram per liter per hour. Corn cob molasses, a hemicellulose hydrolysate containing glucose, xylose and arabinose was used as a carbon source for the P-acidopropionic. Now, the three sugars in the corn cob molasses are simultaneously converted to a propionic acid titer of 71.8 gram per liter with a productivity of 0.28 grams per liter per hour in a fed batch fermentation system with an initial sand density of 14.8 gram per liter. I think you can recall that we have discussed about what is a batch process, what is a fed batch process, what is a continuous process. Okay. So, fed batch process has its own advantages. Okay. The productivity actually increases. In addition to the above food crop residues, non-food crops can also be used as low cost biomass feedstock in the biorefineries. For example, Jerusalem artichoke hydrolysate containing mainly fructose was also used for propionic acid fermentation, achieving a final titer of 68.5 grams per liter, a yield of 0.48 grams per gram and a productivity of 1.55 grams per liter per hour. In addition to the agricultural and food processing wastes mentioned above, crude glycerol as a low value byproduct from biodiesel production has also become an attractive substrate for propionic acid fermentation. Glycerol has a higher reduction level compared to glucose, sucrose and lactate and is thus beneficial to intracellular redox balance and propionic acid production. In general, a high P by A ratio of greater than 10 to 20 can be obtained with glycerol in propionic acid fermentation. High propionic acid production of 106 gram per liter was obtained in immobilized cell fermentation in a fibrous bed bioreactor with glycerol as the sole carbon source. Okay. The fermentation performance with crude glycerol was comparable to that with pure glycerol, indicating propionibacteria was not inhibited by the impurity components such as methanol, fatty acids and salts present in the biodiesel waste. So, this is the best part of this particular research work that is reported by Zhang and Yak, and the reference is given below. Himi et al. In 2000 reported a propionic acid yield of 0.64 gram per gram and a productivity of 0.42 gram per liter per hour from glycerol with P-acidopropionic ATCC 25562 strain. Ruhal and Choudhury also reported the production of propionic acid and trehalose using the P. ferruginariaceae subspecies Sermani and crude glycerol from the biodiesel waste. Liu et al. In 2011 reported propionic acid yield and productivity of 0.57 gram per gram and 0.15 gram per liter per hour respectively from the mixture of glycerol and glucose at a mass ratio of 2 with P acidopropionic ATCC 4965. So, it is a very important work in which actually they use two different types of substrates as a carbon source. Is a mixture of glycerol as well as glucose. Although free cell fermentation of glycerol usually suffer from a low productivity due to the in vivo NADH imbalance, a notably high productivity of 1.44 gram per liter per hour was reached by continuous fermentation with immobilized cells. So, what is being done and reported by Dinisa et al. Is that to balance the NADH inside the fermentation media, they have gone for the continuous fermentation process with the immobilized cells of the propionic bacteria, propionic acid bacteria. So, in summary, abundant low cost renewable feedstocks are available for propionic acid fermentation and they offer an opportunity to reduce the cost of the bio-based propionic acid. So, now we will discuss about the acetic acid, another very important commodity chemical and that is being now manufactured in a large scale from different biomasses. Acetic acid is a colorless and weak organic acid with sour taste and pungent smell. Acetic acid is an important raw material in the chemical industry. The largest industrial application of acetic acid consists of the production of the vinyl acetate monomer, acetic anhydride and various esters. Acetic acid is also used as a solvent in chemical reactions such as the production of the terephthalic acid and the raw material for the polyethylene terephthalate. The global demand for acetic acid is around 6.5 million tons per year, of which approximately 1.5 million per ton is recycled. The remainder is manufactured from petrochemical feedstock or from biological sources. The biological sources, of course, it is less than that of the petroleum chemical stock, but as we have discussed, it has to increase. So, about 75% of the acetic acid in the chemical industry is produced by carbonylation of methanol with carbon monoxide. It is the chemical pathway, chemical process. Only about 10% of acetic acid is only produced by the fermentation, mainly as vinegar. So, let us talk about the microbial production of the acetate. Acetic acid is produced naturally as the sole or main fermentation product by anaerobic homoacetogens and aerobic acetic acid bacteria. Acetic acid bacteria contain 12 genera belonging to the family Acetobacteriaceae. They are aerobic, gram negative or gram variable and ellipsoidal to rod shaped microbes which oxidize ethanol to acetate in a aerobic vinegar fermentation. A high acetate concentration of up to 20% weight by volume can be obtained in aerobic vinegar fermentation with about 60% weight by weight yield from sugar. The process generally involves two steps. The first one is that fermentation of sugar to ethanol by yeast such as Saccharomyces cerevisiae and Clavaromyces fragilis, and second step is oxidation of ethanol to acetic acid by the species of Acetobacter. In contrast, anaerobic homoacetogens produce only acetate from sugar at a theoretical yield of 3 mole of acetate per mole of glucose with an actual fermentation yield of higher than 85% weight by weight. So, table 3 has listed the different types of homoacetogens and their optimal growth conditions and the respective substrates they could degrade to produce acetic acid. So, we will go through one or two. So, Clostridium thermoaceticum. It can degrade fructose, pyruvate, glucose, xylose, lactate, carbon dioxide and hydrogen, even carbon dioxide and hydrogen. So, the optimal conditions is around 60 degree centigrade and the pH is 7. Similarly, there are many Clostridium acetinicum, Clostridium formicoacetum, Clostridium magnum. So, all those can again degrade fructose, pyruvate, pectin, lactate, then malate, xylose, citrate, all these things, and the respective optimal conditions are also given. So, just we can understand that there are many different types of microorganisms are there and they are also efficient enough to degrade various types of substrates at a particular optimal condition. Now, we will talk about butyric acid. So, butyric acid is a 4 carbon carboxylic acid present as an oily colorless liquid that is soluble in water, ethanol and ether. Butyric acid has an unpleasant smell and acidic taste. It is naturally found in butter, Parmesan cheese, vomit and as a product of some anaerobic fermentation. Butyric acid and its derivatives have been widely used in food, chemical and pharmaceutical industries. The acidic form is used to enhance the butter like flavor in food, and the ester form can serve as a flavor in the food and perfume. Butyric acid can also be used to synthesize cellulose butyrate plastics for the textile fiber production. As butyrate can provide protection against colon cancer and colitis, various butyrate derivatives are considered as anti-neoplastic drugs for treating hemoglobinopathies, cancer and gastrointestinal diseases. It is a wide medicinal applications. Butyric acid producing anaerobic bacteria have also been proposed as a novel probiotic treatment for inflammatory bowel disease. Moreover, acids produced from a fermentation process are more favorable than chemically synthesized counterparts for applications in food and pharmaceutical industries. There are many bacterial strains producing butyric acid, which belong to the genera Clostridium, Butyribacterium, Butyrivibrio, Sarcina, Eubacterium, Fusobacterium, Megasphaera, Roseburia and Coprococcus. The genera Clostridium, Butyribacterium and Butyrivibrio are the most studied butyric acid producing microorganisms. However, the preferred strains for potential commercial uses are in the genus Clostridium. The reason is that they are high and stable productivity performance. The next table gives us the butyrate producing Clostridium species with their substrates and the fermentation products. They are gram positive, chemoorganotrophic, strict anaerobes and spore formers. You can see that various types of Clostridium species producing butyrate as the major product. We will just see one or two. The first one, Clostridium aminobutyricum. So, gamma aminobutyrate is the substrate, the products are acetate, butyrate and ammonia. Similarly, if you see and you will come down and see Clostridium barkeri. So, it can degrade glucose, fructose, ribose, mannitol, pyruvate and finally produce butyrate, acetate, lactate, propionate and hydrogen. So, there are many others also there. So, you can later on go through it and have a feeling that what are the different types of Clostridium species been used to produce this one various products such as acetate, butyrate from different types of substrates. Most butyric acid producing Clostridia, they ferment glucose, xylose, lactose, starch and glycerol for the cell growth and butyric acid production. Other substrates such as cellulose and carbon dioxide can also be used for butyrate production. Clostridium carboxydivorans and Butyribacterium methylotrophicum can utilize carbon monoxide and carbon dioxide and hydrogen to produce butyric acid. Clostridium cellulovorans, then Clostridium carboxydivorans, Clostridium polysaccharolyctum and Clostridium populati can use cellulose for butyrate synthesis. Similarly, Clostridium cluiveri can produce butyric acid as a major product from ethanol and acetate as substrates. Most Clostridium species produce butyrate and acetate along with additional products such as ethanol and lactate, but they are in very low concentration. Some Clostridium species such as Clostridium acetobutylicum, Clostridium saccharolyticum and Clostridium thermocellum can be manipulated to form either solvents or acids as the main product depending on the culture conditions. Several species including Clostridium butyricum, Clostridium tyrobutylicum and Clostridium thermobutylicum can produce butyrate as the main product with a relatively high productivity and yield, and they are thus of high interest for potential industrial production of butyrate. The optimal temperature and pH for the Clostridium butyricum and Clostridium tyrobutylicum are 30 to 37 degrees centigrade and 4.5 to 7 respectively. Clostridium thermobutylicum is a moderate thermophile with the optimal growth temperature of 55 degrees centigrade and a pH of 7. Clostridium butyricum is classified into non-toxigenic and toxigenic species. Only non-toxigenic C. butyricum can be used for butyric acid production. Butyric acid bacteria can use a wide variety of carbon sources. To date, various feedstocks have been studied for butyric acid production by fermentation. It is presented in table 5. We will come back to it later. So, butyric acid production from cheese whey, a byproduct from the cheese manufacturing containing mainly lactose and whey protein by Clostridium baezienki at pH 5 gave a final concentration of 12 grams per liter. It is a very good concentration actually, yield of 0.25 gram per gram and a productivity of 0.6 gram per liter per hour. cane molasses, a byproduct of the sugar industry containing approximately 40 to 50% weight by weight total sugars, was used as substrate to produce 26.2 gram per liter butyric acid with a yield of 0.47 gram per gram and a productivity of 4.13 grams per liter per hour. It is a very good productivity. Corn meal after hydrolysis of starch with amylases was used for butyric acid production by Clostridium tyrobutyricum immobilized in an FBB at 37 degree centigrade pH 6, giving a high butyric acid productivity of 6.78 grams per liter per hour and a yield of 0.45 gram per gram and a final concentration of 46 gram per liter. Yes, so we can go through this table which lists some of the butyric acid production from different substrates, the strengths, the concentration, productivity in yield. Let us see the cheese way by using the Clostridium baezerin key. So, the concentration is 12 grams per liter, productivity is 0.6 gram per liter per hour, yield is 0.25 grams per gram. So, if you look at the third one, the cane molasses of using the Clostridium tyrobutyricum, you can see the concentration is 26.2, productivity has increased manifold 4.13. Okay. Similarly, if you look at corn meal and again the same Clostridium species, you can see that productivity is increased manifold again 6.78 grams per liter per hour. Okay. So, these are some of the classical works that are reported by various authors, and the reference, the main reference is given here, Young et al. Okay. So, you can browse it if you are interested more in detail. Okay. So, let us move ahead. So, butyric acid production from wheat flour gave a high final butyric acid concentration of 62.8 grams per liter, productivity of 1.25 grams per liter per hour and a yield of 0.45 gram per gram of glucose. Jerusalem artichoke, after treating with dilute acid to hydrolyze its carbohydrate, mainly inulin, to fructose and glucose, was used for butyric acid production in repeated batch fermentations. The butyric acid productivity, yield and final concentration from the fermentation were 2.75 grams per liter per hour, 0.44 gram per gram and 27.5 grams per liter respectively. Most of the Clostridial species are also capable of fermenting pentose such as xylose. Butyric acid fermentation with corn fiber hydrolysate containing glucose, xylose and arabinose as the carbon source and corn strip liquor as the nitrogen source gave a high butyrate yield of 0.47 gram per gram and a productivity of 2.91 gram per liter per hour. In addition, butyric acid production from brown algae treated with dilute sulfuric acid for hydrolysis of its polysaccharides, mainly the galactan, have also been studied. It is also a very classical work. Okay. These studies demonstrated that butyric acid can be produced from various biomasses including processing wastes and agricultural residues. However, further improvements in the fermentation processes are needed for economic production of the biobutyric acid. So, with this I wind up today's lecture. If you have any query, please register it in the SWAYAM portal, or you can always drop a mail to me at kmohanty@iitg.ac.in. In our next lecture under this module, we will be discussing about 2-3 more such commodity chemicals, 1,3-propanediol, 2,3-butanediol and the PHA. Thank you very much.